Okay, so this is Chapter 1, Part 3. We're moving on to spontaneous generation. This is the belief that basically things can arise from nothing. Okay, so the belief that invisible vital source forces present in matter, I can't talk today, led to the creation of life. So even after the discovery of microbes, many people still believed in abiogenesis, which is spontaneous generation. So even after they found out linked and were able to link, certain bacteria causing certain infections leading to disease, there was still scientists out there that still believed in this spontaneous generation idea. Other scientists advocated biogenesis, saying that living things arise only from others of their same kind. And this has been proven repeatedly over and over again that this is a fact. So a few scientists here. Usually I try to do kind of like a matching for this, but I can't remember what I did for this first test on this. I think it's mostly multiple choice. Louis Pasteur, and that name should kind of sound like something you've heard before, his last name, as far as being related to pasteurization. What did he do? He studied the roles of microorganisms in the fermentation of beer and wine. We like him, right? Swan-Neck Flask experiments were used to disprove spontaneous generation. So what he did was filled flasks with broth and shaped the openings into long swan neck tubes. And this is illustrated in the next slide and also in the textbook. He heated those flasks to sterilize the broth. And those flasks were then exposed to dust from the air. The flasks that were exposed to dust from the air showed microbial growth. The flasks that were exposed to the air but not the dust showed no microbial growth. So when you look at the picture, you can see he broke the flask off at the top so the dust could kind of, dust from the air could settle in easier than they could when the neck was intact. The dust would have to kind of really make its way. The microbes would have to make their way up and in on this side, whereas this side they can just kind of fall in. So he basically helped to disprove that spontaneous generation theory. A couple more scientists related to the microscope. Robert Hooke, he studied household objects, plants, and trees. He described and drew sketches of little structures that seemed alive. Leeuwenhoek, Antoine van Leeuwenhoek, manufactured the first simple microscope, and he studied fabrics initially, and then was able to observe animals, what he referred to as animals in a drop of water. He scraped the surface of the teeth and looked at all the bacteria, or animalcules as he referred to them as, from the teeth. And I guess by the time he passed on, he constructed over 250 small microscopes that could magnify objects up to 300 times. Ferdinand Kohn discovered and described heat-resistant endospores, and we will talk about what endospores are later on in the semester. But just know that endospores are very tough to kill. They're heat, as their name implies, heat-resistant endospores. They're protected in these little capsule-like things that keep them from being destroyed. But he discovered them, and that's an important discovery in the advancements of science. Holmes and Semmelweis describe the importance of hand washing and preventing disease in the hospital settings. I think, if I remember right, they were the ones that talked about going from patient to patient. I think there's a little blip in the book about it, at least in the previous editions there was, where, delivering a baby and then moving on to the next woman delivering a baby and moving on to the next woman delivering a baby, and how disease can spread, infection can spread that way. Joseph Lister used aseptic techniques in surgery. So he's important as well. The germ theory of disease. Louis Pasteur was part of that. Included in inventing pasteurization, he conducted the first studies that linked human disease to infection, and that is germ theory. The idea of germ theory is linking human disease to infection. So you notice how that's bolded and underlined? It's probably kind of important, although generally speaking, I toss out any slides that are not important, but I know specifically that this is a test question. Robert Koch developed the Koch's postulates. These are a series of logical steps that establish whether or not an organism is pathogenic and which disease it caused. So going further than Pasteur did, not only just linking human disease to infection, this Koch's postulates was able to establish a specific organism to a specific disease it caused. He showed that anthrax was caused by Bacillus anthracis in 1875. The year doesn't matter. I'm never going to ask you about the years. Daniel Nathans, Werner Arber, and Hamilton Smith discovered molecular scissors inside bacteria. These are called restriction enzymes. These little molecular scissors allow us to chop up DNA in specific ways and allows scientists to use these enzymes, these restriction enzymes, in tailor-made ways to be able to treat diseases for some genetic engineering, and some other stuff. So basically, it's really important when we're talking about the treatment of diseases. And chances are, if you know somebody that has had a disease, then sometimes these restriction enzymes may have been used to do some research on these diseases. PCR was invented around the 1980s. Polymerase chain reaction, that's what that PCR stands for, and that was a breakthrough in our ability to detect tiny amounts of DNA and then multiply it into large quantities for studying. So multiply or amplify them into quantities sufficient for studying. It has provided new and powerful methods for discovering new organisms, diagnosing infections, and for forensic work such as crime scene investigation. Small RNAs. Genome sequencing has revealed that perhaps only 2% of DNA actually codes for a protein. Much RNA doesn't end up with a protein counterpart. So these pieces of RNA are usually teeny tiny small. And they have found that they have critical roles in regulating what happens in a cell. And all of this information has led to new approaches on how diseases are treated. An example is if the small RNAs are important in bacteria that infect the humans, then they can be new targets for antimicrobial therapy, because sometimes certain microbes are very difficult to treat. So any new ways we can find to treat or prevent infections, then that's pretty important. Genetic identification of the human microbiome. This is not the one that you might be thinking of as far as our genetic makeup. This has to do with the microbiome, the critters that help make us, right? If that makes any sense. All the microbes that are found on us. The first detailed information produced by the Human Microbiome Project was astounding, even though the exact types of microbes found in and on different people are highly diverse. So what somebody in California has on their skin isn't necessarily the same thing that I have on my skin. The overall set of metabolic capabilities that bacterial communities possess is remarkably similar. So they might be different, but they're going to do the same job. This and other groundbreaking discoveries have set the stage for new knowledge of our microbial guests, our little hitchhikers, and their role in our overall health and disease. So this next section, I'm going to stop here and I'm going to pick up on part 4. This next section should be familiar coming over from anatomy. It's a little bit of review of the macromolecules.